Hello everyone, my name is Mikasha Naidu Segaran, SB23088. So what are enzymes? Enzymes are biological catalysts that speeds up chemical reaction by lowering activation energy. They are essential for life and various industrial processes. So now let us see how does enzyme work. The structure of the enzyme plays a major role in its actions. Enzymes are a linear chain of amino acids which give rise to a three-dimensional structure. The sequence of amino acids specifies the structure, which in turn identifies the catalytic activity of the enzyme. So what is the enzyme mechanism of action? Enzyme accelerates biochemical reaction by lowering the activation energy needed for a reaction to occur. They do this by binding to specific molecule called substrates at a region called active sites, forming an enzyme substrate complex. The enzyme then facilitates the transformation of the substrate into a product. After the reaction, the enzyme releases the product and is free to catalyze the same reaction again with another substrate. Let us now look on the importance of enzyme. Enzymes plays a major role in biological processes and industrial processes. In biological processes, enzymes are focused in metabolic reactions and specificity and regulations. So enzymes speed up essential metabolic reactions, ensuring they occur at rates compatible with life. For example, digestion, energy conversion, biosynthesis rely heavily on enzymes. Next, in specificity and regulation, enzymes are highly specific, reacting with particular substrates at the active site, ensuring controlled reaction. This specificity allows for precise regulation of metabolic pathways. While in the industrial process as well, it focuses on enhanced efficiency and various applications. Enzymes can speed up biochemical reactions, leading to faster production rates compared to traditional chemical methods. And also, enzymes are used in food processing, converting raw ingredients into products, textile industries, and pharmaceutical industries for drug development and diagnosis. Enzymes' role in biotechnology and medicine field is very vital in this current generation. Enzymes that are used in this field are DNA polymerase and restriction enzymes such as EcoR1, BUMH1, and HIND3. So these are used for diagnostics and gene editing. While on the other hand, asparaginase is used to treat leukemia. In the biotechnology and medicine field, using enzymes has its benefits and challenges. The benefits are the use of DNA polymerase and restriction enzymes provide high precision, reproducibility, and scalability, making them invaluable in healthcare, agriculture, forensic, and industrial biotechnology. Their roles continue to expand with innovations in CRISP, next generation sequencing, and personalized medicine, while asparaginase is a versatile enzyme with significant industrial value, especially in the pharmaceutical industry for cancer treatment and the food industry for food safety. In the other hand, there is challenges that face to ensure these enzymes produce better quality in biotechnology medicine. For example, DNA polymerase faces key challenges such as low fidelity, cost of high fidelity, variance, inhibition by contamination. While restriction enzyme faces star activity, sequence limitation, methylation, sensitivity, non-in vivo use, asparaginase also faces immunogenicity, toxicity, short half-life, high cost, and food processing stability.
What if I told you there's a natural catalyst working behind the scenes of your favorite bread, cheese, and even your laundry detergent? So, enzyme also working on food and beverage industry. For example, for baking industry, enzyme amylase converts starch into sugars to enhance yeast fermentation and bread volume. Other than that, enzyme protease soften gluten, improve dough handling and shelf life. For dairy industry, enzyme lactase breaks down lactose for lactose-free dairy. And enzyme lipase develop flavor in cheese. For beverage industry, enzyme amylase break down starch for alcohol fermentation in brewing, and pectinase clarify fruit juices and improve juice yield. But if you could produce more food with a better quality at lower cost while protecting the environment, that's the power of enzymes. So benefits of using enzyme, the first one is natural and safe. Enzymes are biodegradable and non-toxic. So enzyme replace chemical additive with natural biological alternative. So when used in food and industry, they can replace synthetic chemicals, example preservatives, bleaching agents, emulsifiers, and also offer a biological process that's gentler and more targeted. For example, instead of using artificial preservatives in bread, amylase enzymes can delay scaling naturally by leaking down starch in a controlled way. Other than that, enzyme also improve processing and efficiency. Enzyme can speed up biochemical reactions and reduce production time and energy consumption. So enzyme play a crucial role that can accelerate chemical reaction without being consumed in the process. Enzyme can improve taste, texture, appearance, and shelf life and help achieve uniform and consistent product standard. Next, challenges or limitations on enzyme. The first one is sensitivity to environmental conditions. As you know, enzymes can lose activity at high temperature or extreme pH levels that can denature and require controlled processing conditions to function effectively. Next, short shelf life. Some enzyme formulations have limited shelf life and require proper storage, for example, refrigeration, dry conditions. Example, lipase. Why short shelf life? Because it loses activity quickly if not stored under ideal conditions, for example, cool, dry, and stable pH. Despite their many benefits of using enzymes, one of the key challenges of using enzymes, especially in large-scale industrial or commercial applications, is the cost involved in producing, purifying, and stabilizing them. This cost factor can limit their accessibility, affordability, and practically in certain industrial origins.
Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Afli bin Muhammad Syed, SB 409. Today, I'm going to talk about how enzymes are used in the production of biofuel and energy. Okay, the first one is the type of enzyme used in the biofuel and energy. Have a three. First one is the cellulase, hemicellulase, then amylase. For the cellulase, we should break down the cellulose, the top fiber found in the plant wall. And the second one is hemicellulase, which will break down hemicellulose, another plant component. And the last one is the amylase, which will break down starch from the grains like the corn into the simple sugars such as glucose and maltose. On this slide, actually, I give you for the diagram, right? Okay, first one is cellulase. Cellulase is breakdown for the plant fiber cellulose. And the second one is the hemicellulase, breakdown for simple sugar that the xylose and arabinose. And the last one is amylase, breakdown for like the starch and the fermented sugar. Okay, for the second is how the enzyme work. And for the pre-treatment, raw biomass is physical or chemical. Treated this break open the top of the plant structure and have the exposed to cellulose and hemicellulose, making it easier for the enzyme to do their job. And the second one is genetic hydrolysis, also known as sanification. Enzymes like the cellulase and hemicellulase break down to complete the plant fiber into the simple sugar. If the starch is present, the amylase convert into the fermented glucose. And the third is the fermentation. The simple sugar produced then fermented by the microorganism like the yeast. This micro convert into the sugar into ethanol renewable biofuel. And the last is the distillation. Finally, ethanol purified and concentrated so it can be used for the fuel. For example, it can be blended into gasoline. Okay, for benefit or use the enzyme, the first one is renewable. We are using natural plant material, not a fossil fuel. And the second one is the lower carbon footprint. Even though ethanol releases carbon dioxide, when burned, that's the carbon is balanced by the carbon dioxide absorbed during the plant growth, creating a closed carbon cycle. And the third is the enzyme work at lower temperature and highly specific, which means we favor byproduct and more efficient sugar production. And the last is help with the energy security, because the country can be produced their own fuel from the local agriculture waste. However, this process is not without the challenges. Okay, the first one is the enzyme production is expensive, especially enzymes like the cellulase, which require advanced method to produce the scale. And the second, plant material as a hard prone to break down the content, the lignin and natural compound that act like glue, making a hard for the enzyme to access the cellulose. And the third is the thermal stability is another issue. Many enzymes don't function with high temperature, so controlling it is necessary, but it's slow the process. And finally, during the pre-treatment, some inhibitory compound release this chemical can block the enzyme activity, reducing the efficiency of the enzyme system.
Real world example, there is lactate milk, product is lactate, enzyme used is lactase. Okay, how it works. Lactase breaks down lactose, the natural sugar in milk, into two simple sugar that is glucose and galactose, which are easier to digest. Okay, the purpose of this is helps individuals with lactose intolerance digest milk without experiencing bloating, cramps, or diarrhea. So the impact is expands dairy consumptions to millions of lactose intolerant consumers worldwide while maintaining taste and nutritional value. Next product is on Casper, enzyme used asparaginase. How it works. This enzyme breaks down the amino acid asparagine, which certain leukemia cells rely on for survival. The purpose of this is used in chemotherapy to treat acute lymphoblastic leukemia, all particularly in children. So the impact is a critical part of leukemia therapy that slows cancer cell growth by starving them of essential nutrients. The pegylations, PEG modifications, increases the drug stability and reduces immune reactions.
Assalamualaikum, my name is Adila Shaira binti Othman, SB 23116. For the conclusion, enzymes are biological catalysts that facilitate important chemical reaction in living organisms. Naturally, enzymes are used extensively in many industries, such as in food and beverage. It helps people every day for creating soft bread and lactose-free milk. Enzymes have been used in medical fields, such as gene editing and cancer therapy. Enzymes can also be used to convert agricultural waste to clean biofuel. The challenge is that they are susceptible to heat and pH challenge, are generally expensive to produce. Any breakthrough in genetic engineering could enable changes in the way we live and interact with our environment. Global challenges: sustainability. Enzymes could optimize waste to energy conversion and reduce reliance on fossil fuels such as renewable energy. Cellulose and amylase can break down crop residues and algae waste into bioethanol. Second, medical breakthroughs. Engineered enzyme may treat resistant cancer with fewer side effects. Asparaginase stop leukemia cell to reduce side effect, and CRISPR-Cas9 edit immune cell to target tumor precisely. For innovation, enzyme engineering using artificial intelligence to design custom enzyme for specific tasks like improving drug delivery, such as AI and machine learning is predict optimal enzyme structure for specific tasks, used to design cellulose that break down plant waste for biofuel. Second, CRISPR enhanced enzyme engineering for precision in gene therapy. Example, use CRISPR-Cas13 by target RNA, allowing temporary gene modulation for viral infection. Third, nanoenzyme was hybrid catalyst that combining enzyme with nanomaterial for target drug delivery or pollution cleanup. It was cheaper, more stable than natural enzyme. Nanoenzyme was used in medical diagnostic to detect cellulose, to detect glucose and cancer biomarkers.